Hello everyone, and welcome to this Shimatsu webinar focused on the analysis of microplastics. I'm Ruth Marfil-Vega, the Senior Market Manager for Environmental at Shimatsu Scientific Instruments. I'll be your moderator today as we talk about our recent work focusing on microplastics. Before we start, a quick couple of notes for everyone viewing. The icons on the bottom of the screen are there to help interact with the webinar, including the help, list of resources, and submitting your questions to the speakers. All of these items are present in your console by default, but if you minimize them or lose them, click the buttons to retrieve them. I want to especially highlight the question and answers area, or Q&A. Please type your questions to the speakers in that box. We will answer your questions at the end of the webinar, and if we run out of time, we'll answer them directly via email. Another important item in the screen is the resources list. We uploaded several documents that are relevant for this topic of microplastics, so you can download them from the links. All right, let's get started. Again, if you are just joining us, I'm Ruth, your moderator. Today we will be talking about microplastics. They are a growing environmental concern. And in the first part of the webinar, I'll be covering some general information about microplastics, as well as the efforts for standardizing the methods in the United States. In the second part of the webinar, my colleague Sudhir will be presenting about the experiments that we did to support these standardization approaches that are leading to harmonized methods for the analysis of microplastics by infrared microscopy. If you want to learn more about the speakers today, Sudhir and myself, please check the bios that are posted in the platform. So let's begin with the presentation. So why are we talking today about microplastics? From a personal point of view, I'm concerned about plastic pollution and how it's increasing everywhere. There were some pessimistic estimates from 2019 that said that the amount of microplastics in aquatic environments was going to be bigger than the amount of fish by 2050. But those predictions have changed because of the impact of the global pandemic and the changes in single-use plastics use, recycling practices. So this is something that not only from a scientific point of view concerns me. From the technical side, we need to find answers to respond to the concerns and regulations that are being developed related to microplastics. And these microplastics are a group of contaminants that are very different from the contaminants that we've encountered in the past in the environmental field. For example, when somebody asks me how do I analyze disinfection by products, that's relatively easy. We know we need to measure the concentration. We use normally a GCMS, and that's it. We have the concentration, question answer. But in the case of microplastics, there are many questions that we can ask about them. What's the size? What's the number? What's the main ingredient, the composition? So today we are going to be focusing in one specific technique that is useful for the analysis of microplastics, and that's infrared microscopy. Before Sudhir presents that work, in the next slide, I'm going to be talking a little bit more about the efforts that are happening in the United States to standardize methods that can help us in answering the questions I just mentioned about microplastics. Researchers from different fields have been working on the analysis of microplastics for several years. But now we are at a key point in this work. Regulations are developing, like what's happening in California, with upcoming regulations for drinking water. There is also the Texas bill to minimize the amount of narvals that are being released into the environment. So environmental stakeholders need standard methods that they can use in response to this regulation, but also what is very important, to compare results from different places, different locations. I need to mention that I'm not representing ASTM in this webinar. I'm a member of the Committee D19 that is focused on water, and I participate in the microplastics efforts. Currently, ASTM is working on several documents. There are a couple of standard practices that have been already approved, and they are related to the sample collection and sample preparation in waters with different turbidity. These documents can be used as reference for collecting and digesting or preparing samples from wastewater to drinking water and all the types of water in between. I mentioned that there are two main topics, but actually there are three topics that ASTM is working on. One was the sample collection and preparation. The second one is the creation of reference materials that people can use to compare the methods and assess the quality of the methods. In addition to the work that ASTM is doing, there are other organizations also working on these topics. ISO is doing a lot of work, and for example, Jennifer Lynch at the Hawaii Pacific University Center for Marine Debris Research has prepared a kit with information from several standards that have been analyzed by infrared and also differential scanning calorimetry. We got one of these kits in our lab, and we are working on generating additional data from these samples. Also, I need to mention that Jennifer Lynch is associated with NIST. So this demonstrates that there is a lot of organizations that are interested in standardizing all the steps in the microplastics analysis. The third area in which ASTM is working on are the actual analytical methods to quantify, detect, assess the composition of the microplastics. There are currently 3 projects ongoing that are going to become 4, and they focus on the use of different instrumental techniques. One of them is focused on the use of infrared. Raman spectroscopy work is going to be separated from the infrared. Then also, there is an effort ongoing for pyrolysis GCMS, and finally, the use of dynamic imaging analysis. When we talk about infrared and Raman, we are normally focusing on counting the particles, assessing the size of the particles, and to a certain extent, also determining the composition of the particles. Pyrolysis GCMS can give you a more deeper characterization of the materials, because you can characterize the main components, but also start studying additives that are in the plastics or other contaminants that are sorbed on the particles. And finally, the dynamic imaging analysis is an alternative technique that can be used for the counting of the particles. Finally, and before Sudhir starts his portion of the presentation, I want to just show you the different analytical instruments that are required to fully characterize the microplastics, depending on the question you have. You want to count them, you want to determine the size, you want to analyze the material. As I mentioned earlier, analyzing microplastics is more complex than other types of environmental contaminants, but once you have clear the question you want to answer, we can help you determining how you have to do that. And with this, I just want to thank you for listening. We'll have time later for the questions, and now Sudhir will start his part of the presentation.
Thank you, Ruth. As we transition to the next part of the presentation, I'll briefly provide an introduction to microplastics and the effect it has caused to the environment. Then, I'll touch on how current lack of standardization is adversely affecting research and advancement in the analysis of microplastics, just to reiterate what Ruth had mentioned earlier. Next, I'll dive deep into some of the specifics of the experiments. There, I'll describe our methodical approach for microplastic characterization, including details, observations, and conclusions. So, just to go over it again, what are microplastics? Well, they are often described as plastic pieces less than 5 millimeters long. They have become increasingly prevalent pollutants, causing adverse effect to the marine ecosystem and other ecosystems that depend on it. These particles enter the environment from manufactured products in cosmetics and other industrial products. In addition, they are also formed as a result of breakdown products of larger plastics already present in the environment. Therefore, the amount of microplastics already present in oceans and other water bodies is alarming. Microplastics are hard to detect or filter out, simply due to their size, and can last for hundreds of years in the environment, often unseen. According to a recent perspective paper, the most pessimistic estimates suggest that by 2050, the mass of microplastics in aquatic habitats will surpass that of fish. As Ruth mentioned earlier, with the heavy increase of single-use plastic in the last year, and as we continue to use them more and more, this could very likely happen earlier than 2050. It is well known that microplastics is an environmental problem that adversely affects coastlines and marine ecosystems. Although it is a big unknown at this point, microplastics have the potential to affect human health as they've been found in seafoods. Microplastics eaten by larger marine animals generally pass through their bodies. However, research has shown that microplastics can be retained in the guts for extended periods where they may cause abrasion and damage to internal tissues. The smaller particles, or nanoplastics, can pass through the gut walls and travel to different organs such as lungs, livers, and kidneys, where they can cause damages. A lot of research is ongoing, and further research is required to understand the full potential health implications from ingesting microplastics. So, where are we now in terms of even beginning to solve the problem? The first step, which currently is the primary focus of most research works, is to identify and characterize the particles in terms of their size and other physical properties. A recent review published in Trends in Analytical Chemistry provides an excellent summary of various techniques suitable for identification of microplastics and their scope. However, despite efforts focused on identifying, quantifying, and assessing the size of microplastics in the environment, standardized methods are lacking. As a result, studies cannot be compared or reproduced easily. As it is well known in the field, infrared microscopy has become an essential technique in the toolbox for the analysis of microplastics in the environmental samples. In this work, we conducted a methodical evaluation of significant operational parameters of infrared microscopy for accurately determining the size of microplastics, with the overarching goal of outlining performance parameters that will help in the standardization of the analysis. The aim of our work was to solve simple problems that are often overlooked but are significant to help standardize the analysis of microplastics. first thing we investigated was the filter material to see which one gives the best possible sensitivity and data with infrared microscopy. The filters are commonly used to filter out microplastic particles from aqueous samples. Then we looked into how microplastics can be identified and mapped with the help of an infrared microscope. Finally, in the most important part of the study, we determined the threshold size of plastic identification. Although instruments specification lists smallest possible measurable dimensions, measurement of smallest possible sample in a practical setting may be different. Here, we will present two experimental approaches backed by statistical analysis to determine the threshold of measurable sizes in practical setting, reasonable time, and with standard measurement parameters. In order to determine the best filter material, we tested three types of commonly used filters to filter microplastics: cellulose ester filter, glass fiber filter, and stainless steel mesh filter. All three types are commonly used to filter microplastics from aqueous samples. Our sample consisted of 20 micron diameter polystyrene beads in deionized water filtered through three types of filters. After the samples were filtered and left to dry on the respective filter materials, they were put under infrared microscope objective for spectral measurement. Reflectance spectra from the polystyrene beads on each filter materials were collected with identical measurement parameters. As seen here, the spectrum of the microplastics generated on cellulose ester filter was very noisy. Although some background subtraction can be done to improve the signal-to-noise ratio, heavy absorbance of infrared by cellulose ester would still adversely affect the quality of signal. Glass fiber filter produced very good data. However, it was quickly apparent that the particles could quote-unquote hide between the glass fibers, making the measurements very challenging. Stainless steel mesh filter produced the best data with the least amount of noise. Since the steel reflects almost all infrared beam, it is an excellent background material that helps produce highest signal-to-noise ratio among the three filters. Therefore, we highly recommend the use of stainless steel mesh filter for microplastics filtration. Next, we investigated distribution of microplastics in a given area. Instead of simply looking at one particle, the infrared microscope was set to take spectral measurements to map the presence in an area. Using software algorithm and search functionality, unique spectral characteristics were used for identification and mapping. The mapping technique is very powerful as it can provide a bigger picture of the distribution of particles. An example of measurement of 100 micron diameter polystyrene sample is shown here. Left is the visible image with measurement grid superimposed. Right is software mapped image superimposed on the visible image. Here, red denotes higher concentration of particles based on prominent peaks found in polystyrene. Orange, yellow, green, and blue subsequently mean lower prevalence of polystyrene. Similarly, on this slide, 50 micron size polymethacrylate particles are shown. The left picture shows the visible image of particles and right shows relative prevalence of the particles as determined by the infrared microscope. One more example. On the left, slightly hard to view are the polystyrene particles, only 20 microns in diameter. And on the right is the relative prevalence of the particles as determined by the infrared microscope. Although pure particles are shown to help better correlate the visible image with the mapped image in these examples, multiple types and sizes of particles on a single piece of filter can be analyzed at any given time. With the help of the software, multiple species and sizes can be identified simultaneously. Switching gears to our next part of the study, we carried out experiments to understand the smallest size of microplastics that can be measured using infrared microscopy in a practical setting. Theoretically, the spatial resolution of an infrared microscope is determined by the combination of diffraction limit and detector element size. For infrared microscope, based on the operating range and diffraction limit, the limit of spatial resolution is about 5 microns. Our measurement parameters included typical parameters that are often used in infrared microscopy. The measurement range was set to mid-infrared with resolution of 8 reciprocal centimeters. All measurements were carried out in reflectance mode with 15x reflectance objective mirror, and most importantly, only 64 scans per measurement point was averaged to keep each measurement under a minute while obtaining high signal-to-noise ratio with a single-point MCT detector. It was important for us to use these typical or commonly used parameters for understanding the smallest measurement size because these settings would give a practical value, which is often larger than theoretical or factory-specified values. In order to determine the smallest measurable particle size, first, the infrared microscope was set up to measure in reflectance mode. We focused on one single microplastic of size of 100 micrometers. Then, we set up the measurement aperture of 5 microns by 5 microns to obtain infrared spectrum from that particle. Subsequent measurements were taken for the same particle with larger apertures at 10 by 10 microns, 20 by 20 microns, and so on up to 100 by 100 microns. The increasing apertures allowed us to simulate measurements of varying sample sizes while maintaining all measurement conditions and eliminating sample-to-sample variations. Then, peak-to-peak signal-to-noise ratio were determined at each size and plotted. The graph in this slide shows signal-to-noise ratio versus particle size. As expected, signal-to-noise ratio increased as the size of the particle increased. Next, we plotted the difference in increase in signal-to-noise from one measurement size to another. This is shown by the blue plot. Here, interestingly, we found that the greatest increase in signal-to-noise ratio occurred when the sample size was increased from 5 microns by 5 microns to 10 by 10 microns. This finding suggests that the threshold is somewhere between 10 and 5 microns. So 10 microns can be used as a safe measurable threshold size. As the data demonstrated exceptional increase in quality of signal when the size is increased from 5 microns to 10 microns. To further confirm the measurable size threshold, another study was carried out with sample size of 5 by 5 microns and 10 by 10 microns. In this study, the number of scans averaged to obtain a spectrum were gradually increased. The number of scans were set at 32, 64, 128, and 256 for the two sample sizes mentioned below. Measurements were replicated 5 times and signal-to-noise ratio determined at each size. The idea is that the increase in number of spectra averaged should increase the signal-to-noise ratio if the signal is real. As shown in the graph on the right, signal in the 5 by 5 micron sample size did not show any substantial improvement with the increased number of scans. The average signal-to-noise ratio values stayed close to 0.5, so it is safe to say that significant real signal was not observed at this size. When the sample size was changed to 10 by 10 microns, signal-to-noise ratio showed a noticeable increase with the increase in number of scans. Theoretically, as mentioned earlier, signal-to-noise ratio should increase as more scans are averaged when the signal is detected. Therefore, this result further verifies that the signal from the 10 by 10 microns was real and that from 5 by 5 microns was primarily noise. Therefore, this confirms the threshold of 10 by 10 microns. In conclusion, we presented a series of method and experimental conditions in infrared microscopy for identification and accurate determination of size of microplastics. Furthermore, we have demonstrated that by using typical operating conditions of infrared microscopy, it is possible to identify and distinguish between microplastics particles as small as 10 by 10 microns. This threshold value was determined and verified by two separate analysis methods, each supported by statistical analysis, therefore further verifying the capability of the technique. Finally, the results presented in this paper are a subset of data being prepared for standardizing the analysis of microplastics in environmental samples. If you would like to learn more and get detailed experimental information, please check out our paper published in the August 2020 issue of Spectroscopy Online. This concludes our presentation for today. I thank you for your time and attention and hope that the presentation was helpful. To learn more about our efforts on microplastic analysis and work on other environmental issues, please visit our website onelab1earth.com. To learn more about infrared spectroscopy, spectrophotometers, microscopes, and use of these instruments for applications like microplastics and others, please visit fdirlab.com. With this, I'll hand it over to Ruth for Q&A session. Thank you.
Thanks, Sudhir, for the presentation. And thank you to our audience for attending and sending the questions. There's been several that came through the chat, and please keep on sending more questions through the Q&A box. Now we'll answer your questions. We've received lots of them, and Sudhir and I will be taking care of those. The first question I have here is for Sudhir. Your data, especially the mapping, shows one type of plastic standard per analysis. Have you tried analyzing multiple plastic species at a time? If so, how complicated is the analysis? Sudhir?
So in the images we've shown here, for the purpose of clarity and to avoid confusion, we've only used the one type of plastic or one sample per image. As discussed earlier, the main purpose was to quote-unquote see the particles of different sizes. However, we did measure and mapped samples with more than one type of plastic particles, as long as there is at least one unique peak or one unique area in the infrared spectrum for each type of particles. Mapping them and displaying all of them in one single image is very easy to set up in the software. All right, actually I see a question that is probably better answered by Ruth. So here it goes. Is there a formal established definition of microplastics?
Thanks, Sudhir, for that question. As mentioned during the seminar, microplastics are a very diverse type of contaminants, and there are several parameters that can be measured. There has been a lot of effort across different disciplines and countries to try to standardize a definition of microplastics. And the one that I like to use as a reference is the one that was established by the California Water Board in July of 2020. It's very specific for drinking water, but it's one of the first official definition of microplastics. It's long, so what I'm going to do is I'm going to paste the link to the definition in the chat. But basically, it defines microplastics as particles between 1 nanometer and 5,000 micrometers. Okay, we have the next question, and this time is for Sudhir. The question says, I was curious how hard it is to search for a single particle, say of about 20 micron in size under the microscope.
Okay, I can see how this would be challenging when one would try to find micron-sized particle under the high magnification of measurement optics. In our case, the microscope was equipped with what we call the wide field camera. This camera takes a high-definition image of about 1 centimeter by 1 centimeter area, and once that image is acquired, it can be zoomed to locate the smaller particles, and then that image itself can be used to help move the stays. In other words, if you see, once you highly magnify it, you simply double-click in an area, and then the stays will move to that area to bring the particle or that area under the view of the measurement objective. So instead of trying to look for a very small particle in a live feed, a high-definition image is used, which makes this process really super simple.
Thank you, Sudhir, for the answer. Let's see the next question. Where can I get the published paper? I'm going to take this one, and I'm assuming the question refers to the paper that Sudhir talked about in his presentation. The paper was published in Spectroscopy, but also we've added the link to this paper in the resource section that you can find in the platform. In addition to the link to the paper that we talked about today, there are other few relevant items that you can download related to microplastics analysis. And here is another question for Sudhir. Sudhir, can you go over of your size confirmatory study where you use multiple number of scans to verify that 10 by 10 microns can be used as a safe, smallest measurable threshold value?
Sure. So what we did as we were planning this experiment was basically said to ourselves that if the signal is real, averaging more of them should increase the signal to noise ratio. Again, this is true only if the signal is real. So we first used 5 micron size particle and averaged different number of scans. The signal to noise ratio, with the increase in number of scans, stayed pretty flat, clearly suggesting that all or almost all of the data we were getting was noise, or there was no significant signal, real signal. On the other hand, similar study with a 10 micron particle showed that the signal to noise increased as the number of scans were increased to obtain the average. As we know, theoretically, this only happens when the signal is real, therefore clearly showing that the practical threshold value in this case was somewhere between 5 to 10 microns. So in our case, 10 microns can be safely considered as a good threshold value. All right, on to the next question for Ruth. What are the steps required for sample preparation before the analysis of microplastics by infrared?
Thanks, Sudhir. The sample preparation depends on the amount of solids in the sample. So it's going to be different whether you are planning on analyzing a drinking water sample or an effluent from wastewater or wastewater influent. A large component of the organic matter in samples with high suspended solids is cellulose. As Sudhir mentioned during his presentation, this chemical has a stroke absorbance in the infrared spectrum, and the peak can cover the peaks from other compounds of interest. Then the key aspect of the sample preparation is going to be to remove cellulose. Per the guidance created by ASTM, samples are filtered through different sizes sieves. Then the content from each size fraction is going to undergo a wet peroxide oxidation followed by enzymatic digestion. There are other approaches using different chemicals, but they all follow the same overall workflow. If you are interested in more details about the specifics in the ASTM method, please send me a message and I'll be happy to share that information with you. Next question is for Sudhir. Although I have not used infrared microscope as a lab person in a water analysis lab, my understanding is that the higher-end FTIR microscopes to look at micron-sized particles are expensive. Are there lower-cost alternatives? And if yes, what are the limitations?
Thank you for the question, Ruth. Yes, you are correct. The study here uses our high-end microscope. It has liquid nitrogen cooled detector, that allows it to measure low micron level sizes, and it is on the higher end as well. However, there are alternative systems. There is a smaller microscope, in fact, that uses slightly less sensitive room temperature detector. The basic rule of thumb is that if you are looking for particles 60 microns or larger, this would be an excellent tool for you. It does not have some of the automatic measurement functions, but the data quality itself for 60 microns and larger is actually very good. For smaller particles, though, the higher end system is a must. For more larger particles, say 200 microns or larger, individual particles can be analyzed in FTIR spectrophotometers itself using an attenuated total reflectance accessory, or more commonly known as ATR accessory. This is another alternative that is also lower cost.
Thanks for giving us an overview of different alternatives for analyzing microplastics by infrared. The next question is also for you. We all know microplastics persist in the environment. So the question that came through the chat says, how can we distinguish new particles from others that have been in the water or other environmental samples for a long time?
Thank you for the question. As you know, identification using infrared is dependent upon how good our unknown samples match with the species already in the library. As we all know, most libraries out there have spectra of pure or clean species. In other words, all run with ideal conditions. If there's a new particle or naturally whether particles are being measured, in most cases the spectral match will not be as good or will be incorrect. However, such natural conditions can be mimicked in the lab to artificially degrade the plastics, and then their spectra added to the libraries. In fact, we have recently come out with a plastic analyzer package where we've included spectra for ultraviolet degraded and thermally degraded plastics under different conditions. Actually, we have included a paper in the resources sections for that, so please take your time to go over it. In addition, I can think of the old-fashioned way to get around this to some extent. One can analyze or look into only a part of spectra or certain peaks to get more understanding of the unknown. Finally, with newer softwares, there are solutions available that can aid in identifying unknowns in a sample if information about one or two more species are fed into the software solutions.
Thanks, Sudhir. I just one quick follow-up question. You mentioned that you have libraries that are built with UV and thermal, that the libraries have been built based on results from thermally and ultraviolet degraded plastics. My question is, can customers update the libraries and make it their own and add their own entries?
So the short answer is yes, but let me elaborate. The libraries that are included by Shimazu's, they are what we call is protected, just so that it was built to a certain specification and is there as a complete package that is provided. However, creating your own libraries and making the software look into your and our libraries together at the same time is super simple. Once you have your spectra, it literally takes a couple of clicks to add those to the libraries.
Thanks, Sudhir. I think you brought a very good point. The quality control that was put into the making of the library is something that we cannot forget.
Yep, definitely. All right, I see another question that is, I think, very much suitable for Ruth. So here it goes. I am a water quality supervisor at water utility, and I'm new to IR, or infrared. How difficult is it to get started with the analysis of microplastics by this technique?
Thanks, Sudhir, for the question. Implementing the infrared analysis may sound very challenging because it's not common. Infrared is not commonly used in environmental labs. And we know, as we talk today, microplastics have their own challenges related to the diversity that they present. But I come from the mass spectrometry world. I've been doing mass spectrometry analysis for different types of emerging contaminants and other contaminants in water for many years. And what I can say is that operating at the infrared is less complex than operating other instruments like a GCMS that are routinely used in environmental labs for the analysis of volatiles, for example, or operating an LC-MS for the analysis of PFAS. So it can be challenging at the beginning, but day-to-day operation is less complex. Also, to help you with implementing this application, Shimadzu offers free familiarization after the installation, as well as free phone and email tech support for the life of the instrument. And this is something, this support is not provided by the engineer that goes to fix the instrument. There is a team of scientists that are there to answer your technical questions. So just keep in mind that there are two different groups. The group of engineers that fixes the instrument when the instrument is broken, and the scientists that are there to help you with the practical application. Also, probably if you're in a water utility, you probably have been using Shimadzu's TOCs for a long time. So the latest software releases for both instruments are give you the opportunity of connecting the TRC and the infrared through Lab Solutions database or client server platform. So that way you can network the instruments and streamline the data processing and managing the information from your samples. So yeah, just to summarize, it might be challenging from the beginning. You might think it's going to be difficult, but at the end, the operation of the instrument is less complex than other instruments commonly used in environmental labs. So don't be afraid and start working on the microplastics analysis. Well, looks like we are nearing the end of our allocated time for the webinar. Thank you all for the questions. Sudhir, do you have anything else to add before we close the event?
Sure. I just wanted to stress that the webinar is available to view on demand with the same link that you've received in the email, and we'll also post it in our website. Thank you.
The questions that we then answered during the webinar will get back by email to the person who submitted it. And once again, thank you all for attending and participating. We hope to have you back in upcoming webinars we organize in the future, and have a great day. Thank you so much for listening.